Hello everyone, I am Sutapa, MSc in Biology. Welcome to my channel, Sutapa's Biology. My today's topic is nucleus. You know from the lower classes that nucleus is the brain of the cell. That means it controls all the metabolic activities in a cell, and as it control all the activities of the cell, so naturally in a total organism also nucleus controls everything. So nucleus is the brain of the cell, and today's topic is nucleus. It was first discovered by scientist Robert Brown on 1831. Robert Brown discovered it on 1831, and from that time it was believed that each and every living cell must possess a nucleus. But later in some cases we have seen that it's not true, because there are many cells in plants and animal which do not have this nucleus also. Now I am giving the example of that animal cell. In case of the animal cell, most of the mammals erythrocyte means the RBC, do not have the nucleus, and in plant cells you can see that the sieve tube, it has also no nucleus. So without nucleus also some cells are there. The sieve tube cell in plant without nucleus. Note it carefully. And in nucleus, it's what are the parts of the true nucleus? That's the main thing. Nucleus is the true nucleus. If the nucleus is true, then only you can call it nucleus. Otherwise, in case of the prokaryotes, you have seen they do not have the true nucleus. That's why their nucleic material is known as nucleoid or genophore. So here, what are the parts of the true nucleus? Coming to that part. We can see the true nucleus. True nucleus in eukaryotic cell only, not in prokaryotes. And in other words, nucleus is known as karyon. The nucleus is known as karyon. So the division of nucleus is known as karyokinesis, and in every parts of the nucleus we shall get the word karyo sometimes. Now nucleus has first, number one, it has nuclear envelope. It has two parts, that is it's another words known as karyotheca. It's known as karyotheca. Number two is nucleoplasm. It's also known as karyoplasm. Number three, nucleolus. And number four, nuclear reticulum. Nuclear reticulum. So these are the four main parts of the true nucleus, and firstly I am discussing about the nuclear envelope. It's a double membrane bound structure, and in a cell generally we can see the single nucleus, but sometimes in some lower group of organisms like in fungi we can get two nucleus in a cell, that's known as dikaryotic cell, and sometimes we can get many nucleus in a cell without any separation, that's known as sinusotic cell. Sinusotic cell, I am giving the idea about that. In case of some fungi, the nucleus are many in a single cell without any separation, and that's known as sinusotic cell. Sinusotic cell. Suppose in case of the fungal cell, they have the hypha-like structure containing many nucleus together. And this is known as, these all are nucleus. These dots are all are nucleus and there is no separation in between, and this is known as sinusotic cell. It's we can find in Rhizopus, Mucor, etc. These are fungi. Now, in case of animal cells also, some cell they possess multiple nucleus without any separation. That is known as syncytial cell. Syncytial cell. We can get it in striated muscle. In our striated muscle. So, multiple nucleus is present in case of sinusotic cell of plants and in case of syncytial cell of animal. Other than that, in some fungi, dikaryotic cells are found also which contain two nucleus. In other cases, that is the normal case, we can see that the single cell possesses single nucleus. Now coming to that nuclear envelope, first part of the nucleus. It's a double membrane bound structure and it's known as nuclear envelope. It's a double membrane bound structure. It has exokaryotheca, the outer wall, and endokaryotheca, that is inner wall. So these two layers form the double membrane bound structure nucleus. Now I am giving a rough diagram of the nucleus here and then coming to the other topics also. It's a round structure, round to ovoid sometimes, mostly round. So like this, it has two membrane. That is outer is known as exokaryotheca. These are RER, connected with RER. So, outer layer is connected with the RER. So, these are dotted. RER is ribosomal. These are dots of ribosomes. Rough endoplasmic reticulum which contain the ribosomes outside. So, this is the outer membrane, and inside the nucleus there are many nuclear reticulum. In an interphase nucleus they remain like the lump of the wool and in this manner, not identified as chromosomes. These are the nuclear reticulum or chromatin. Okay, this has a very prominent nucleolus, sorry, it has a very prominent nucleolus. So inside the nucleus, the matrix part is known as nucleoplasm or karyoplasm. This is exokaryotheca. This is endokaryotheca. This gap in between these two is known as perinuclear space, perinuclear cisternae or perinuclear space, perinuclear cisternae or space. Perinuclear space. This is chromatin reticulum or nuclear reticulum. Nucleolus, and nucleoplasm. Okay, you are seeing all these parts. Roughly, I have given the diagram of this, and if there are two envelopes, that is nuclear envelope, exokaryotheca and endokaryotheca. In between the two envelopes, they have the perinuclear space, and inside it, chromatin reticulum or nuclear reticulum, nucleolus, and nucleoplasm. So this is about the rough diagram of the nucleus. Now I am coming to the details. That is nuclear. Another thing is there. This is the nuclear pore. These are the nuclear pores. Nucleus remain within the cytoplasm, we know, and if there is no connection of nucleus with the cytoplasm, then nucleus will be an isolated system and that of no use. So it has to be connected with the cytoplasm scientifically, and for this connection they need the nuclear pores, and this nuclear pores, they form another thing, that is nuclear pore complex. Later I am coming to the discussion of nuclear envelope. I have already told you that the nuclear envelope has two layers, and in between them there is the gap that is known as perinuclear space or perinuclear cisternae, and after that nuclear membrane has some pores which are distributed throughout the nucleus very regularly, nuclear pores, and these pores are regulated by the structure that is known as annulus. The nuclear pores and annulus in combination, they form the nuclear pore complex. Nuclear pore complex, and that's guarded by diaphragm at a structure that is known as diaphragm. So this is about the nuclear envelope. And so nuclear envelope has these parts: exokaryotheca, endokaryotheca, perinuclear cisternae or space, nuclear pores plus annulus by diaphragm. They are nuclear, forming the nuclear pore complex. So this is about nuclear envelope. Now I am coming to the nuclear reticulum. The main important thing of nucleus is the nuclear reticulum, as it forms the chromosome, and the chromosome contains the DNA, which is the blueprint of our body. So DNA is the blueprint of our body, of every organism, if they like that. Lump of wool. Now nuclear reticulum is made up of chromatin fibers. Chromatin fibers which condense to form the chromatid. They condense to form the chromatid, and after that the chromatid condense to form the chromosome. Now the main thing, that's DNA is present in between the chromatin fibers. DNA, the functional part of DNA, we know that that's the gene, and that controls our all characteristics. So this is the main reason why the nucleus is known as the brain of the cell. This is due to presence of DNA inside the chromatin fibers. Now chromatin fibers, they have different types. Two types are there. One is euchromatin, another is heterochromatin. Chromatin fiber has two types broadly. One is euchromatin and another is heterochromatin. Now what is euchromatin and heterochromatin? In lower classes you have not studied, so this is the new word to you and very important for the competitive exam as well as board. So what's the euchromatin? In cell division we can see there are two stages, that is interphase, that the preparatory stage of the cell division, and another is divisional phase. And telling about this or mentioning here about the cell division, as the chromosomes are visible only in the cell division. In the dividing cell we can see the chromosome, so I am giving the reference of the cell division. Now in cell division there is the interphase, when some parts of the, if we can see under the microscope, electron microscope, then we can see that some parts of the chromatin fibers, they are very condensed and taking very dark stain. They are taking very dark stain. They are very condensed also, coiled. These are heterochromatin and these are not functional too much. And another is light colored, that is the light stained region. And this is the euchromatin. The heterochromatin are genetically less active, but euchromatin are genetically more active. Why? Because here in heterochromatin there are the repetitive sequence which are condensed to form inactive region, and in case of the euchromatin, their chromatin fibers, they contain more DNA and they are more genetically active. So due to presence of less, why less active? Less DNA. Why more active? More DNA. So this is about the chromatin fibers. And in dividing cell, again I am telling about the dividing cell, as we can observe the chromosomes under dividing cell. So in a dividing cell, in mitosis we can get prophase, metaphase, anaphase and telophase. All these are the subphases of the divisional phase mitosis. Now in case of anaphase, we can see that the chromosomes are moving towards the pole, and during this movement they take some shape. According to that, chromosomes are divided into many types, like metacentric, submetacentric, actocentric and telocentric. Acrocentric, there is a sequence, as you can remember it well. Acrocentric and telocentric. All are given in your NCERT book. Metacentric means it has centromere at the center. What is centromere? Which connect the chromatids with each other. This is the centromere. If the centromere is at the center and two arms are equal, then it's known as metacentric. And during the movement, it will take the shape of V. It's the V. Then submetacentric, it has longer arm than the other. It has some longer arm than the other, and during movement, chromosomal movement, they will take the shape of L, that is submetacentric. Then acrocentric, their one arm is very longer than the other, and during chromosomal movement in anaphase, they will take the shape of the eye. And another is telocentric, which has only one this is J, and then this will take shape of J, that is the cursive J. And telocentric, it will take the shape of the eye. It has only one arm and centromere at the terminal end. Centromere at the terminal end, and it's known as eye. So metacentric, submetacentric, acrocentric, and telocentric, all these are categorized on the basis of the chromosomal movement during anaphase of the mitotic cell division. Now about this nuclear reticulum, and next we are coming to the next one, that the nucleoplasm, that is the matrix region. It contains many nuclei, many enzymes, many minerals, many mineral ions, and etc. Many more things are there, as it's the place of all the synthesis. So it's known as nucleoplasm or karyoplasm. And last one is nucleolus. Nucleolus is a very dense particle, round particle inside the nucleus, and it's very prominent. Some cells contain more than one nucleolus, and some cell contains single nucleolus. Nucleolus is amembranous. It remains within the nucleus without any membrane. That means its density is more than the nucleus, so it's remain isolated in the nucleus without any membrane system. Now nucleolus has some parts. It was discovered by Fontana. Scientist Fontana discovered it, and discovered nucleolus. And in a chromosome, it remains attached with the in chromosome, the inward, is known as inward, where the nucleolus remain attached. Nucleolar organizer region. Nucleolar organizer region is the origin of the nucleolus. Origin of the nucleolus. Now nucleolar organizer region, where it's located, is located in the secondary construction of the chromosome, of the SAT chromosome. Later I shall tell about the SAT chromosome in my following videos. Here I am just telling that it's the inward is on the secondary construction of the SAT chromosome. The centromere is known as primary construction, and other than that, other constructions are known as secondary construction. So nucleolus remain attached with the secondary construction of the SAT chromosome, and it has some parts. Nucleolus has some parts, that is pars amorpha, pars granulosa, pars fibrilla, and perinuclear chromatin. So nucleolus number 1, pars amorpha. Number 2, pars granulosa. This is the granular part. Amorpha is the amorphous part. Pars amorpha, pars granulosa. Number 3, pars fibrilla. Pars fibrilla. It's the fibrillar part. And number 4, perinucleolar cisternae. Perinucleolar chromatin. Perinucleolar chromatin. The chromatin fibers which are connected with the surface of the nucleolus, that's known as perinucleolar chromatin. As we know, then inside the nucleoplasm they remain with the nucleolar reticulum. So surrounding nucleolar reticulum of the nucleolus is known as perinucleolar chromatin. Other than that, it has a amorphous zone, it has a granular zone, it has a fibrillar zone. So according to that, these parts of amorphogranular, the fibrillar and perinucleolar chromatin. Now, what's the function of the nucleolus? The nucleolus has a very important function, that's the rRNA synthesis. I think you are not yet introduced with this rRNA, that is the ribosomal RNA. Ribosomal RNA. The ribosomal RNA forms the ribosomal subunits. So, ribosomal RNA forms the ribosome. If we break the ribosome, then you can get the ribosomal RNA, and this ribosomal RNA, they are synthesized inside the nucleolus. So this is very important for the function. It has very important function in a cell. So this is about the nucleus, and just giving some hints about the function. Number 1, it's the brain of the cell, so control all metabolic activities. Number 2, it has nuclear pores through which it can control the nucleocytoplasmic traffic. Number 3, it has nucleus which can synthesize the ribosomes. Number 4, nuclear reticulum is the main thing for the DNA, and it can transmit the characteristics from one generation to other in the living organisms. So these are all about the nucleus in short. So that's all for today, and okay, bye.